Well, thanks very much. And I have to say, I'm staggered at the outburst of interest in immunity. I need to check first that you are in the right talk. And there is no shame in leaving at this stage. I have been slightly annoyed if people leave halfway through, but I mean, at this stage, if you're in the wrong talk. I've also really struck talking to people as Kevin. It's fantastic to see so many friends from many different aspects of my professional life, but I'm also a bit daunted by that because I realise I'm talking to an audience which runs, and I don't mean this in any order, but which runs from a sort of an eminent professor of medical history to sort of one of the world's leading immunologists on HIV and everything in between. And it's quite difficult in a way to present this kind of talk to such a wide audience. But what I'll try and do is avoid any jargon. But one thing I'm really happy for people to do is to interrupt. So if you feel I've slipped into using sort of impenetrable jargon, which I will try not to do, please ask me to clarify, because there's nothing worse than sitting, or actually there is something worse than sitting not understanding what's going on, and that's standing not what's going on. So interrupt me if it's not clear. Just a brief few slides to introduce. I realise not everybody in the room is focused on malaria all the time. Malaria remains a global problem. By global, we mean global around the world, anywhere within the tropics. And this kind of map shows its distribution, but it doesn't really give an indication of its load, if you like. That's easy to see in these kind of projections, which take account not only of the amount of malaria, but the number of people. And from that, you can see that malaria is essentially a problem of Africa and Asia, and particularly India. And that's because there's a lot of people in India. But around 90% of the total mortality due to malaria occurs in Africa. And that's what most of my talk is going to be about. It's based on work that we've largely done in Africa, but it would apply to malaria wherever it exists. As a reminder to those people who don't think about malaria every day, you get malaria when you're bitten by a mosquito, an Anopheles mosquito. I should explain, by the way, that if you see me sort of keep coming back, I like to walk around when I'm talking, but I've been told I've got to stay near here because apparently it's been recorded. So I will forget that from time to time and drift off, and then I'll suddenly dart back again. You get malaria when you're bitten by an Anopheles mosquito. The mosquito has picked up the malaria from biting somebody else a few weeks earlier. It picks up a stage of the malaria parasite from the blood. It undergoes a kind of complicated process in the mosquito, which is not of particular interest to us today. And then it injects this sort of beast, highly magnified, a sporozoite, which finds its way quickly to your liver. There's a liver cell full of parasites. And during all this stage, you're blissfully ignorant of the fact. So this isn't malaria disease. You've got a parasite, but you're not diseased. The disease only occurs when the parasite comes out of the liver into the blood, and there's a parasite entering a blood cell, and there are parasites in stained blood cells. And this is the one bit of the cycle that we're interested in when it comes to human disease. And it causes a range of manifestations, which would range from a relatively mild, sometimes asymptomatic infection, to at the other end of the spectrum, what we're really concerned with, which is a devastating fatal illness. A whole range of different manifestations, but essentially historically one of the world's major killers. A lot of what I'm going to talk about today is based on the fact that people do become immune to malaria. And this is obvious, and it was obvious to Robert Koch over 100 years ago, and he made this point that if you are in a malaria endemic area and you look at who's suffering from malaria from the point of view of illness, not from the point of view of being parasitized, it's children. Clinics are full of children with malaria. By the time you become an older child or an adult, you're essentially not troubled by the risk of dying of malaria. You may still have malaria, as I'll go on to indicate, but you essentially are out of the period of risk of dying of malaria. So you certainly become immune to malaria. And what's often said about it is that it's slow to develop, it's incomplete, and that we don't understand it. And I've been guilty of saying these things, both in talks and in proposals, particularly in proposals. You always say we don't understand it because you're trying to persuade people that you need to do more work on it. But I've got a bit bored of saying these things, partly because, well, the last one is a bit sort of, looking back on 30 years of work, I'm rather embarrassed to say we don't understand it. But actually, I don't think any of these things are strictly true. And I'm going to try and convince you that although this is how immunity is often characterized, it's actually not strictly true. Malarial immunity is slow to develop in some senses, but not absolutely. It is incomplete, but it's pretty good in many senses. And actually, I think we do understand quite a lot about it. I'm going to try and convey some of that in the talk today. And then at the end of the talk, I'm going to take a step back for a few slides and look at what the overall impact of malarial immunity is and what it will be as malaria is controlled. Why are we interested in immunity to malaria? There are two reasons, really. The prime one is that we really would like a vaccine against malaria. I think most people accept that it won't be possible to eradicate malaria using current methods, although current methods, it is possible to get much better control than we've currently got. And I think it's important to recognise that you can control malaria with current methods, but I don't think anybody thinks we can really get rid of malaria without extra tools. And probably, historically, vaccines have been the most effective public health intervention against any infectious disease. There is another reason why I think we should be interested in immunity to malaria, and it's one that often isn't given much emphasis, and it's one that I suppose some people would challenge, but at the end of the talk, I'll give some evidence to support this. And that is that malaria can be argued to have been a major determinant of the human immune response in general, not simply the immune response to the malaria parasite, but as a major determinant of our immune response to pretty much everything. And I'll come back to that towards the end of the talk. So this puts into a sort of figure, as it were, the picture that you saw of children and adults in the malaria endemic area, and tries to sort of put a bit more understanding on that. On the y-axis is just amount. It doesn't matter what the scale is, it's just amount. This is nothing, that's a lot. And this is the age of a population in the malaria endemic area. And shown on it in different colours are different indications of immunity to malaria. Now, I need to be clear here that when I'm talking about immunity, I mean acquired immunity. I don't mean immunity in the sense that some of us may be resistant to some things because of our genetic makeup or whatever. I mean the process by which, by experiencing a pathogen, we develop the ability to limit the growth of that pathogen. Everyone's familiar with this concept from childhood illnesses. Perhaps the best example of things like chicken pox. When you have chicken pox, you get all your kids to go around and get chicken pox because you know that when you've had chicken pox, you won't get it again, at least not in that form. Malaria is rather different to that, and shown here are different manifestations of malaria. The one to focus on first is the pink outline. So this is the age of the population, and this is the proportion of the population who are parasitized at any one time. So if you just go and take a slide from them, they've got parasites in their blood. And you can see that certainly by this index, malaria immunity is very slow to develop. You don't even really begin to show any until you're in your 20s, and it's never complete. You're always parasitized. And it may be more so than this. This may be an artifact of our detection systems. It may well be that if you were able to look sensitively enough, that actually in a malaria endemic area, most people have got parasites most of the time. So in that sense, if we were saying that's our index of immunity to malaria, then it definitely takes a long time to develop. It's definitely very inefficient. But at the other end of the spectrum, in green, is shown that, if you like, that manifestation of malaria, which from a public health point of view, we're most concerned about, and that's severe malaria. Malaria where you have a distinct chance of dying as a result of it. And there you see a quite different dynamic. This is a problem in young children. This varies with the amount of malaria, and I'll say a little bit about that later. But to a first approximation, in most malaria endemic areas, you don't see malaria deaths beyond the age of around about 5 years. You still get malaria, you still get this thing called mild malaria, which I won't talk about, and you're still parasitized. But from a very important point of view, you've acquired the ability to limit the parasite to in a way which is very important to you. You don't die. So in that sense, it's not true to say that malaria immunity is totally inefficient. It's a pretty important thing to be able to avoid dying. And it's slow, but it's not that slow compared with some of the other manifestations of malarial immunity. Because of the fact that malaria isn't like chickenpox, you don't just get it once and then you're immune, the way we examine how you become immune to malaria involves having to look over a period of time. And this schematic shows the kind of studies that we do. You basically start at a point in time and you follow groups of individuals, children or adults, over time, and then there's an end point, and during that period of time you either will or won't get malaria. Now, from now on, when I talk about will or won't get malaria, I mean you'll get malaria the disease, not the parasite, although the same concept could obviously be applied to being parasitized if that's what you're interested in the immune responses to. But we're interested in what is it that limits the ability to get disease and then to die from malaria. And over that period of time, what you'll find is some people get malaria and some people don't. And we just call those who do for that period susceptible and those who don't relatively protected. Now, even this kind of approach gives, I think, a wrong sense of what's actually going on, and you get a much better sense if you look at some real data, and these are data from a cohort of children that we've followed for a long time in East Africa, in Kenya, and this is showing so this represents a group of nearly 400 children followed for 6 years, and this is the number of malaria episodes, so clinical episodes of malaria experienced by those children, by each individual child over time, and this is the proportion of the population that experienced that number of episodes. So 10% of that population didn't have a single malaria episode over the entire 6 years, and they're living in exactly the same place as this few percent who had 20 episodes. So when you get down to the detail, even within a small area where everybody's exposed to malaria parasites, there's enormous heterogeneity in people's experience of acquiring immunity. Presumably, there's something about these children which enables them to acquire immunity with relatively little ill effect. They may experience none or 1 or 2 overt episodes of malaria. And at the other end of the extreme, there's a group of children, it's not a small group, it's about a third of the population, who experience a lot of malaria. And we know that it's this group who are particularly susceptible to severe malaria and to dying of malaria. And this isn't just random, that's the Poisson distribution indicator there, i.e. if you threw all this data at the wall and saw how it looked, that's how it would look by chance. So this isn't a chance effect, and it's happening in a population where they're all exposed to the same level of malaria. So there's something about a significant proportion of the population which makes it difficult to become immune to malaria. But on the other hand, there are children who do seem to become immune relatively easily. And I think that's the thing that you miss out if you take a general description of how difficult it is to become immune to malaria. So again, just to remind you of the cycle, when it comes to a human response which would limit the parasite, there are several points where this could take place. You could attack the parasite as it's injected, as it were, from the mosquito into the bloodstream before it gets to the liver. You could attack the parasite in the liver, or you could attack the parasite in the bloodstream. Now, I'm not going to talk at all about the bits going between the mosquito and the liver. That's not to say it's not important, and in fact it's very important from the development of malaria vaccines. The best candidate vaccines actually target this stage. But we don't believe that this aspect of immunity is very important in naturally acquired immunity. As in all these things, you always have to kind of slightly eat your words. We said this for many years. Recently we've been looking at this in a bit more detail. It may well be that there is a response here which is important, but we don't lose much if we simply say that for natural immunity, this isn't particularly important. Similarly, you can develop an immune response to that stage of the parasite which is transmitted to the mosquito. This wouldn't protect the individual, but it might prevent transmission of the parasite, and that's also a good target for vaccination. But that's not what I'm going to talk about. I'm going to talk about the parasite in the blood which causes disease and what kind of immune responses people naturally develop to that. One thing we know about immunity to the blood stages of malaria is that it is conferred to a large degree by antibody. Now, antibody is that soluble component, as it were, of the blood which we make, it's molecules we make which attack organisms which allow them to be cleared from the bloodstream. It's often contrasted with cellular immunity. The cells in the blood are the things which do the damage. So everyone, I think, well, those at a certain age in the audience will be familiar with antibody in the sense that before there was hepatitis B vaccine, if you were going to somewhere where there was a lot of hepatitis B, you used to have to go and have a really painful injection. And the injection was just antibody collected from people who got high titers of antibody in their blood, and it was kind of purified, injected into your leg, and it lasted you 6 months and it stopped you getting hepatitis B. Fortunately, that's not necessary anymore. But the same principle applies to many infections which are controlled by antibodies. So that's a soluble component, as it were. And we know that antibody is important from studies done over 50 years ago, which is sort of illustrated briefly here. So this is a group of children with malaria parasites who are treated with various formulations of antibody, including antibody from non-immune Europeans, and including a group who are given antibodies from adults living in a malaria endemic area. And you can see a very dramatic, essentially getting rid of the parasites very quickly once you've given antibody from people who've grown up in an endemic area. So we've known for many years that antibody is really important. It doesn't mean that other things aren't, but it does mean, and for various other reasons, we believe antibody is most likely to be the mechanism by which you control blood stage malaria. So I'm going to talk about antibody and identifying the potential targets and the ways in which it works. If we look at the blood stages of the parasite in more detail, here's a malaria parasite attaching to a normal red cell, and it's about to go in. Once it's in, sort of you roll forward sort of 24 hours or a bit more, it actually modifies the red cell enormously. So if you go to this side, these are normal red cells, nice round smooth red cells. That's a one with parasite in it, and it modifies the red cell enormously. Later on, the individual daughter parasites break out, and so that little round thing there is the same as that round thing there. It's about to break out and start the cycle again. This is a bit puzzling in a way. You would think, having gone to the trouble to hide inside a host cell, which is normally a way of avoiding immune responses, you would think that you wouldn't bother to modify the surface of the host cell and make yourself a flag, as it were, to the immune system. And presumably this is because it has to, the parasite has to. It serves some function. We know some of the functions these modifications serve, and they serve to stick it to other cells which protect it from the immune response. But by modifying the surface of the cell, they put a flag on it and say, this is not a normal cell, and it becomes a target for the immune system. I'm going to talk about antibody responses to both these stages. This stage, the merozoite entering the red cell, and then this stage, the modified red cell. And although it would be logical to talk about this first, I'm actually going to talk about this first, partly because of my own interest historically that was the order it went in. If you put blood containing these parasites, if you take the parasites out and put them into the serum from individuals who have been exposed to malaria, antibody sticks to these modifications, and you can tell that by, if you look under the microscope, you have to imagine this is a film under the microscope. All these rather faint things are normal red cells, and these clusters are parasite cells. And the reason they're clustered is the antibody has bound to the surface of the modifications, and it's locked them together. So that's how we know, and that's how we often test for, are there antibodies which can stick to these targets on the infected red cell. Many years ago, I particularly was interested in whether or not you make a response and what is the nature of that response. And these data show something really quite important about that response. So seen here, along the top are individual children who presented with an attack of malaria. Along the side is that individual child's acute and convalescent serum. That's to say their serum taken at the time they presented and their serum taken 3 weeks later when they'd had chance to make a response to the parasite. And the thing to take from this is that there is a very evident diagonal. That is to say, each child comes in, hasn't got a response to the parasite which is causing disease, and then 3 weeks later has. But in general, although there are exceptions, most children don't have antibodies to all the other parasites. This is really quite a striking finding because it implies that all parasites are different, or at least many parasites are completely different, and that you make a response to the parasite you've got, but maybe not to other parasites. Another way of looking at that, to illustrate exactly that, is shown here. Here we've mixed 3 different parasites. We've taken parasites from 3 different children, 3 different parasites. So what I've just said is that maybe all the parasites are different. And they've been put into a single plasma from an individual, and we've coloured each parasite differently. So there are 3 parasites here. There are red parasites, blue parasites, and green parasites. And you can see that in fact those clumpings do show that the response is to very specific individual parasites. It's not that you make a response to the parasite and that's what you've got and it's good. What you've actually got is a response to each different sort of parasite. Now this has kind of led to an idea of the so-called hole-in-the-wall hypothesis. And the idea was that maybe you had to acquire all these different specificities in order to become immune to malaria. And there is actually quite a lot of evidence that that does happen. Now, shown here is a particularly striking single instance of that. This actually is the same sort of experiment, and again, along the top, not labelled because it would make the slide messy and complex, are lots of different parasites tested against a first and second sample from the same child. Now, in this case, it's a little bit different because this is a very young child whose antibodies actually came from its mother. It wasn't antibodies that the child had made, it was antibodies from the mother. And what you can see is the one parasite which actually caused the disease in this child was the one parasite to which that child had no antibody to start with. and then within a few weeks had developed very strong antibody to it. So this is, if you like, a graphic illustration of the idea of a hole in the wall. You build up a repertoire of responses, but you don't have all of them, and the parasites which cause disease are the ones that slip through the gaps in your own defense. We subsequently looked at this in a much larger study of children, of older children, and I won't go into the details of this, but confirmed that that is what happens to some degree, that once you develop a response to these very variable antigens, you actually don't then get disease with parasites carrying those antigens. That's quite interesting, but it's rather depressing from the idea of how that would inform, say, vaccine development, because this is an extraordinarily variable system. At one point we thought maybe it's infinitely variable. We now think there are probably around about 60,000 distinct antigens on the surface, or versions of these antigens. So it would be impossible to imagine that you either become immune to malaria by having to experience all of them, because there's no way you could experience all of them in a given period of time. It would also be impossible to imagine developing any sort of vaccine approach based on that degree of variability. So it's a bit depressing. But in fact, it turns out that you do also make responses, eventually, some people, which cross-react, which break down that variability, and which can actually say, no, this is a parasite. I'm not bothered whether it's parasite A, B, C, or D. And this is illustrated here in a rather beautiful picture. Here's a big clump of parasites which have been clumped by putting them into serum. And it's the same three parasites I showed you before. There's red, greens, and blues, but this time they're all clumped together. And that shows that the target which is being recognized is not a very variant target. It's a target which is shared between all parasites. Now I have to be clear here and say we don't know how common this response is in relation to the very variable responses, but at least it does indicate that it's possible to break that variability and eventually to recognize common targets. And then if you look at this in the kind of cohort study, which I indicated earlier, where you look at this response, you follow children over time, and shown here, and this is a so-called Kaplan-Meier plot, where you're looking at a whole group of children followed over 200 days, and this is the chances of going down with malaria disease. And you can see that those children without this broad antibody quickly develop malarial disease, whereas those who've got reasonable levels of this kind of antibody response are relatively protected. Some of them do develop episodes, but far less commonly than that group without antibody. So at least with regard to these antigens on the surface of the modified red cell, there is evidence that as you develop a response over time, you begin to see beyond the variability and develop a common response, and that response does to some degree protect you from disease. I'm going to change now and look at this stage of the parasite, the merozoite. This is the only point at which the parasite, as it were, is free and completely exposed to the human immune system. It's a naked parasite, it's not hidden by a cell, and it wants to get into the red cell as quickly as possible. It actually does this probably in about 30 seconds, so it's a quick process. But this kind of process, for most infectious diseases, you would guess would be exactly where the immune response would target, because if you can break this essential step, then obviously you abort the whole life cycle. And that's true of many viral diseases, and so it's an obvious target for an immune response. In more detail, that stage of the parasite is actually pretty complex. I mean, the malaria parasite is a lot more complex than viruses and bacteria. I mean, it's more like thinking about, if you talk about immunity, it's more like thinking about developing immunity to cancer cells than it is to, say, viruses. It's a pretty complex cell. It's got a very complex surface, and it's got all sorts of bits inside, all of which can be involved in this process of getting into red cells. And we've just labelled a few of them here. But the basic message is there are lots of components of the merozoite which could be the target of an immune response. This horrible slide is not meant to be looked at, but to illustrate a particular point. And that is, over 30 years or so, lots of groups looked at some of these targets on the merozoite and said, do immune responses to these targets protect you against malaria? And the result was an absolute dog's dinner. This was reviewed by Freya Folks a few years ago. There had been 33 of these kind of studies. And a couple of important points. Firstly, although there had been 33 studies, only 5 antigens had ever been looked at. And we think there are probably about 5,000 proteins in the malaria parasite. So you're looking at a tiny proportion of the total proteins. And the other thing that came out of this was the results were all over the place with the same antigen. Sometimes, so this is the line of no protection. Some studies showed good protection, some showed none, some showed useless protection. So, I mean, a good summary at that stage, perhaps 5 years ago, of 30 years' work for groups all over the world would have been, well, actually, is not clear at all. I mean, there is no clear evidence. And it was really by thinking about this kind of result that we and others began to think, well, maybe we're thinking about this in the wrong way. And I think there are two ways in which we weren't really thinking about it very clearly. And these are so obvious that when I say them now, I'm embarrassed, because I think anybody who's not an immunologist in the audience would say, well, that's so obvious. I can't believe that you sort of wasted time not thinking of that. But that's the way it works out. The first obvious thing that we really didn't take much account of for many years was the amount of antibody you had. So illustrated here is the idea that if this is the amount of antibody to anything that you have, then going from low to high exposure to that infection, you develop more antibody. And that there is probably a threshold that you need for protection. Now that threshold might be different for each antibody response you've got, but the important point is that there is a threshold. So if you just simply say, have I got this response or not got this response, then you're going to mix up poor responses, which simply reflect having been exposed to a pathogen, with good responses, which reflect having had the right kind of exposure. So that's a sort of, if you like, a general model. And from that, you would predict that if you look at any given response, sometimes you will find that they're not related to protection, and sometimes they are. And then this is exactly what we did find when my colleague Faith Ossier looked initially at a study. It looks complicated, but it's actually quite, I'll just talk you through this diagram. This, again, is in a cohort of children followed over time. What this reflects is the chance that a child would develop a malaria episode. So naught means no chance of developing malaria. 0.7, there was a 70% chance of developing malaria. In the cohort as a whole, 35%, that's the red line there, developed malaria over the time. So that was your chance overall of getting malaria. And then shown in the different colours are responses to different components of the parasite, the antibody response to different components. And it's asking the question, this is amount of antibody. So as you get more antibody, do you become protected? And a couple of things jump out from this. The first is, and it doesn't matter what these antigens are. It probably does matter to the people who spent their career working on them, but it doesn't matter at all in general. The thing that steps out is that some things, it doesn't matter how much antibody you've got, it barely modifies your chance of getting disease at all. These are probably not protective responses. But other responses, there's a very clear relationship between the amount of antibody you've got and your chance of getting disease, such that if you've got a little bit of it, you've actually got an increased chance of getting disease, which probably just reflects the fact that you're being exposed to malaria. But as you cross a certain threshold, and that threshold is different for different responses, you actually increasingly have less and less chance of getting disease. So a couple of things from that. Firstly, amount does matter. And secondly, more than one response appears to be protective. And that's the second thing, which I think if you asked any non-immunologist, you'd say, well, obviously, if you've got a complicated organism, it's not going to be one thing, it's going to be lots of things. But again, the way I guess people tend to think about things is they focus on the thing they're interested in. And the change which I think has happened over the last few years is that we've stepped back and said, well, actually, it's probably that you have to build up not repertoires against a single antigen, but you probably have to build up repertoires of responses which additively give you protection. And this is illustrated in this slide, which again is from Faith Ossier, which shows two separate studies. So these are two different groups of children followed over time. This is the chance that they were protected from no chance to 100%, i.e. complete protection. And this is the number of responses they made which were above that threshold point. And it doesn't matter what the response was. So here we've said, let's ignore what the response is. We're not bothered. It's not what we used to say, which is, oh, you've got to have a response to this, you've got to have a response to this. It's just saying, let's just give a score. If you've got a response, call it 1. If you've got 2 responses, call it 2. And what you see is that as you build up your responses beyond a certain point, you actually get more and more protected, to the point that if you've got 5 or 6 of these responses, those children are actually completely protected against malaria. So here, I have to remind you, we're talking about an age group of children who we would traditionally say are susceptible to malaria. They're having trouble becoming immune. But in fact, within that group, there are individuals, even at that stage, who are already completely protected. And they're the ones who've got a good collection of the right responses. Now, actually, the merozyte is much more complicated than that. And there are probably a couple of hundred potential targets on the merozyte. And I told you earlier that we've actually only looked at 5. Historically, people have only looked at 5. They didn't do that because they were lazy. They did that because it's difficult to produce these antigens in a purified form which you can study. Now it's not so difficult, it's still not trivial, but we've recently been collaborating particularly with Kevin Wright, and again this is Faith Ossia's work largely, Kevin Wright at Sanger, to produce highly purified, fully functional individual antigens from the surface of merozoites. It doesn't matter what they're called, this is all, each line is a separate antigen, so these are all the little different components of the merozoite. Each one is different. And shown here is the proportion of an immune population of children who do one of 2 things. The blue is the proportion who simply have evidence of a response, an immune response. So it's saying, do you recognize this bit of the parasite? The red is the protective efficacy of that response as determined in the way that I've just shown you. And a couple of things jump out from this. The first is that lots of children respond to lots of things. That's not a surprise. The second thing, though, is that some of these responses, and these are responses, remember, to antigens which we've not looked at previously, some of these responses are actually apparently highly protective. And what's quite striking is that those that are highly protective are relatively rare, whereas those that are relatively common tend not to be very protective. And there's this kind of evolutionary sense in that. You can understand how that might happen. If you apply the same sort of approach which I've just talked about, that is, instead of saying you've got to have this response or that response, but just say, well, no, what you've got to have is a collection of good responses. And that's what's shown in this little box here. So here again is the protective efficacy. There's 0. A minus efficacy would imply that it was actually bad for you. And there's 100% protection. That means you just don't get malaria at all. So this is applied to a large cohort of children where these measurements have been made. And if you just take any one antibody response, that's the levels of protection which you largely get, up to about 30-40%, with a couple of outliers which you can't see buried away in there. If you take random collections of these responses, just randomly pull out of these 36 or so, just take any random combination of 5, this is the distribution of protections you get in black. It gets better, so having more than response is better than having one response. But if you take random combinations of just 5 picked out of the top 10 in this group, this is what you get. Again, you approach pretty much 95 to 100% protection. So what I'm saying is that these children, there's a whole group of children in the cohort who actually are apparently fully protected against malaria, and what those children have, which is different from other children, is that they've got collections of the right responses. They've got good responses, and they've got more than one of them, and it doesn't matter which one they've got, so long as they're good responses. It simply adds up to place you in a position where you're much less likely to get malaria. Okay, so what I'm going to do now for the last 5 minutes of the talk, having sort of gone through what, if you like, what the phenomenology of immunity is, who it affects, and the time rate at which it occurs, and tried to convince you that actually immunity to malaria is probably largely the combination of specific responses added up in the right way in the right individuals. I want to now take a step back and say, what's the overall importance of immunity at a population level? And this will become particularly important as we control malaria. I think many of you will be aware that malaria is being reduced in many parts of the world, and particularly in many parts of Africa. This is what's actually happening in Kilifi. So Kilifi, for those of you who don't know, is there. That's where we work, on the coast of Kenya. This is from 1990 to 2009. This is just a measure of the amount. It doesn't matter what the scale is. And this is severe malaria in hospital. And you can see that when we started working in Kilifi, and that's why we started working there was lots of malaria. Now, there's very little malaria. This actually isn't because of control. That's when bed nets became widely available, and this is when good drugs became widely available. You can see there's a really interesting phenomenon going on here, which is that malaria has been going down for a long time. But that's a separate talk, but it's worth kind of knowing that it's not just control which is doing this. But the point is that malaria, which used to be a massively important determinant of childhood mortality in East Africa, or at least on the coast in East Africa where we work, has become a relatively rare disease. It's extraordinary. It's really extraordinary to work in an area and see that over a period of years. At the same time, if you look at overall childhood mortality in the same area, so this is the childhood mortality rate, the death rate per 1,000 child years, that's 1990 up to 2010, that's also plummeted. This is really absolutely remarkable. People often talk about how depressing it is that Africa's not going to meet its Millennium Development Goals, and that's true in general. There are many parts of Africa where there have been absolutely massive reductions in childhood mortality, and it's gone along with the reductions in malaria. But obviously that's not cause and effect. So the question is, to what degree has the drop in malaria caused the overall reduction in childhood mortality? And this looks rather complicated, but it's not too complicated, and it's really worth, I think, understanding it, because it's got a massively important message, and it's in two parts. This figure is the same as the kind of figure I just showed you. It's basically, but now it's from 1999 to 2007, and instead of showing just malaria, malaria is the red line going down dramatically over that period. It's also showing, and you can ignore all these other lines, look at the blue line. The blue line is all invasive bacteremic disease. Now, what I mean by invasive bacteremic disease is severe bacterial disease, meningitis, severe pneumonia, septicemia. And this is actually the thing which kills children. In all parts of the world, when you have high mortality, no matter how important malaria is, what's much, much more important is bacterial disease. All added up together to a first approximation, what kills children are bacteria, not malaria. I mean, I say that having sort of put the argument around for ages that malaria is really important, which it is, but bacteria are much more important. And that's what's happened to bacterial disease over the same period of time. Now, again, you've got the same problem. You say, well, something's changing, and it's all going in parallel, but maybe it's just changing socioeconomic circumstances, maybe it's better access to healthcare. But there's a very, very strong reason to believe that that's not the case, that actually these are absolutely causally related. And this is shown in this figure, which I'll explain. I think most people will know that there's a very interesting story about sickle cell disease and sickle cell trait. So sickle cell trait is a genetic change which affects populations, particularly in malaria endemic areas, or populations which historically came from malaria endemic areas. It's a genetic modification which, if you just carry the gene, has no obvious deleterious effect at all, and in fact, has a very positive effect. It protects you from severe malaria. And that's the reason why it's grown to large proportions in populations exposed to malaria. The downside is if you have two genes, it causes a very unpleasant disease, sickle cell disease, which has a high early mortality rate. So it's a balanced polymorphism where the gene rate increases because it's protective against malaria, but it can't increase too much because the eventual loss by the chance of having two together outweighs the protection you get from malaria. Now, we've known that for years. We know that sickle cell trait is protective against malaria. But a few years ago, we did a study where we looked at whether sickle cell trait was protective against anything else. And to our surprise, we found that sickle cell trait was strongly protective against invasive bacterial disease. Nothing to do apparently with malaria, just protects you against invasive bacterial disease. Now, there are two reasons why that could be. It could be genuinely protective by mechanisms which you don't understand, or it could be that malaria itself is the risk factor for developing invasive bacterial disease, because we know sickle cell trait protects against malaria. And we spent ages trying to work out how you could work that one out, and we had all sorts of study designs which involved going to look in parts of the world where there was no malaria and such like. And then the penny suddenly dropped with us that while we'd been thinking about this, malaria had gone right down in Kalifi where we were sat. So we went back, and this is largely the work of Anthony Scott, Tom Williams and other colleagues, and we looked at, so here there's the same scale, it's running from 1999 to 2007. This is the malaria coming down, and each year we performed, using the large-scale surveillance data that we have, a case control study to say, does sickle cell trait protect you against bacterial disease? Not against malaria, against bacterial disease. And in fact, as we'd originally observed, it was strongly protective. So that would make no protection at all, neither one thing or the other. That's the line of neutrality. And the prediction was that if sickle cell trait works against bacterial disease by working against malaria, then as you have less malaria, the protective effect against bacteria will disappear in the population. This is a natural experiment. It's a natural Mendelian randomization experiment. And this is exactly what happened. You can see the protective effect, which is very strong against all forms of bacterial disease, simply disappears as malaria disappears. unless somebody in the audience can come up with one, I think there is no other possible explanation for these data other than it is malaria which leads to invasive bacterial disease. So if that is the case, and because one can work out the rates, you can estimate that malaria not only causes a massive toll of mortality in terms of malaria, it actually causes a bigger toll of direct mortality by its immune suppressive effects in making you susceptible to other diseases. Now the problem is when you get to the end of that kind of line of argument, you begin to get a bit sort of, yeah, but that's what academics do. You know, you add one thing to another and they say if this then that, and you make a great big story about it. Is it really believable that malaria is that important that it causes most childhood mortality in endemic areas? Well, actually I think it is. And I think some really good data to illustrate that. This is Bioko, which is the sort of island part of Equatorial Guinea. And Bioko has had historically horrifically high childhood mortality rates. Shown here are the years 2000 to 2007. This is the childhood mortality rate, not malaria mortality, this is childhood mortality rate. And when it says 157, it means that for every 1,000 children born, 157 of them die before their 5th birthday. So these are horrific childhood mortality rates. In Bioko, they implemented a malaria control program from 2003 onwards. So here is each year's childhood mortality rate over that period of time. For the period leading up to the control program, it was 157. For the period afterwards, it had been taken down by 2/3 to 55. But the really striking thing about this is this wasn't a gradual process. This all happened in 1 year. Sort of with a really radical malaria control program, it was possible to reduce the total childhood mortality in the whole country by 2/3 by doing one thing, and that was controlling malaria. And I think, again, this cannot reflect changes in social behaviour or anything else because it all happened in a very short period of time with a single intervention. So I think there's a really dramatic demonstration that it really is the case that malaria not only causes malaria deaths, but it leads to many other deaths. Now the relevance of this to malaria immunity is that immunity to malaria doesn't only protect you against potentially malaria deaths, but it may well protect you against other things too. And the relevance of that is that as we reduce malaria, which is a good thing, we also reduce immunity to malaria. So I showed you these slides earlier on showing the population and becoming immune to malaria. Now as you increase the amount of malaria transmission, this all contracts across to the left. You force all the disease down into very, very young children. You have a high mortality in young children, but you acquire your immunity quickly. As you drag it out to the right, if you reduce transmission, you stretch all these things out, and in fact you stretch them out in a way which isn't illustrated here, which this green line begins to stretch right the way across. So the ultimate stretching out will be most of us in this room, i.e. people with minimal malaria exposure, where everybody's susceptible to dying of malaria, with a high chance if you get infected and you're not treated. So push it one way, you push it all into children, drag it the other way, you send it into an older population. Well, obviously what we're doing by controlling malaria is exactly this. We're reducing transmission and we're dragging the whole immune curve across to the right. Does this really happen? Well, in fact, we've had a very dramatic demonstration that this is what is happening recently. So here's what I showed you before, severe malaria in Kilifi, dramatic reduction from 1999, massive amount, 2009, 10 years later, hardly anything. But if you look at this now in a different way, the red line on each figure is exactly the same, and it's the averaged out amount of malaria throughout the year from January to December for the whole 20-year period. Now I showed you 1999, same shape but somewhat above the average. That was a really bad year. By 2009, hardly any malaria, way, way below the average for every month of the year. Suddenly, in 2011, for 3 months of the year, we had a massive epidemic which banged right back up to the historically high levels. The difference was, this was in older children, much older children, very high death rate in children aged 5 to 10. Suddenly our wards, having been empty of malaria for 5 years, were full of malaria again. It was a very dramatic demonstration of the danger of what happens when you lose malarial immunity in a population. This is happening because we're stretching the curve across to the right. Now the argument here is not that controlling malaria is a bad thing. Obviously it's not that. controlling malaria is a really, really good thing. That's the point I've been making about the importance of malaria in relation to childhood mortality in general. But as you control malaria, one thing you will inevitably lose is the population immunity to malaria. And what that means is that if you ever lose control of malaria before it's eradicated, you've got a whole population now at massive risk, children and adults, of returning back to epidemic malaria. Now, not only affecting young children, but affecting all ages. And that would be an absolute disaster. And this isn't a theoretical risk. This has happened many times in history. It happened in India and Sri Lanka in the 1950s and 60s, and it happened in Madagascar in the 1980s, where you get massive return of malaria with very high death rates in all ages of the population because you've lost your immunity to malaria. So, in summary, what I've tried to convey is in two parts. Firstly, naturally acquired immunity is not always totally slow and totally inefficient. It's really pretty good at some things. Antibody concentration, obviously, I mean, it is obvious when you think about it, is really critical. There are many, many more targets than we've previously thought about, and what's probably important is the combinations of responses to the key targets. It's not any single target. So obviously from that, the key questions now are, well, what are the best combinations of responses? Which responses could you induce which will give you that 100% protection? And why is it that it's a relatively, it's actually not a massive minority, but why is it that many children take much longer? There's something obviously about some people which makes it more difficult, and that, to understand that will be pretty important in trying to develop an approach which involves increasing people's immunity. And then at the very last part of the talk, I've given a bit of a warning that however slow and inefficient malarial immunity is, it's really, really important to the population level. It's what stands between very high death rates in older children and adults if malaria resurges, and that resurgence is a real risk if you don't keep malaria control absolutely. So the message of that is there can be no relaxation at all in international commitment to controlling malaria. Once having gone down that route, which is a good thing, it's absolutely essential that it's continued, because if it's not continued, the abolition of natural immunity to malaria will produce a disastrous situation. I've drawn on the work of lots of people in giving this, and I've shown many of our group here. Particularly want to acknowledge Faith Ossia and my colleague Faith Ossia and our joint students Kathona Kamuya and Linda Murungi, and many collaborators around the world. And to thank you once again for inviting me to give this talk. Thank you very much.